In the early 1960s, Italian scientist Ferruccio Ritossa made an unexpected observation while studying fruit flies. He noticed that exposing the flies to a slightly higher temperature caused a peculiar change in their cells. Distinct puffs indicating increased activity appeared on their chromosomes. This intriguing phenomenon, triggered by heat, hinted at the existence of a hidden cellular response. These puffs turned out to be regions of DNA actively producing a specific type of protein. This protein, later named heat shock protein, HSP, was produced in large amounts when cells were subjected to stress, such as heat shock. Ritossa's discovery opened up a new avenue in biology, revealing that cells have sophisticated mechanisms to cope with environmental challenges. The implications of this discovery went far beyond fruit flies. Scientists soon realized that heat shock proteins, or HSPs, were present in virtually all organisms, from bacteria to humans. This widespread presence suggested a fundamental role for these proteins in basic cellular processes. Ritossa's serendipitous finding paved the way for decades of research into the fascinating world of molecular chaperones, the unsung heroes of the cellular world. Heat shock proteins, or HSPs, are a specialized group of proteins with a critical job, protecting the cell from damage. They are produced in larger quantities when the cell experiences various forms of stress, including heat, cold, or exposure to toxins. Think of them as the cell's emergency response team, ready to act when things get tough. But HSPs are not just about damage control. They also play a vital role in the everyday life of the cell, particularly in the complex process of protein folding. Proteins are the workhorses of the cell, carrying out a vast array of functions. But to do their jobs properly, they need to fold into precise, intricate, three-dimensional shapes. This is where HSPs, acting as molecular chaperones, come into play. They assist in the folding process, ensuring that proteins adopt their correct shapes and remain functional. Without chaperones, proteins could misfold and clump together, leading to cellular dysfunction and potentially disease. Imagine a long, tangled string. This string represents a newly synthesized protein. For this protein to work correctly, it needs to fold into a specific, intricate shape. This folding process is a bit like solving a complex three-dimensional puzzle. While some proteins can fold correctly on their own, many require assistance. This is where molecular chaperones, like HSPs, step in. They act as guides, preventing incorrect folding and helping proteins navigate the complex folding pathway. Chaperones provide a safe environment for proteins to fold. They bind to exposed parts of the protein, preventing them from interacting with other molecules and forming harmful aggregates. Think of chaperones as chaperoning the protein folding process, ensuring that everything goes smoothly. Without chaperones, the cell would be in a constant state of protein misfolding chaos. Proteins would clump together, forming useless and potentially toxic aggregates. This can disrupt cellular processes, leading to a range of diseases. Among the many types of chaperones, GroEL and GroES stand out as remarkable examples of molecular machinery. Found in bacteria and chloroplasts, these two proteins work together to assist in the folding of other proteins, particularly those that struggle to fold independently. GroEL and GroES form a large cylindrical complex that acts as a protected chamber for protein folding. Picture it as a cellular life raft, providing a safe haven for proteins to fold correctly amidst the crowded cellular environment. Section 6. A helping hand. How GroEL and GroES guide protein folding. The GroEL-GroES complex functions like a sophisticated molecular machine, using energy from ATP hydrolysis to assist in protein folding. GroEL forms the cylindrical chamber, while GroES acts as a removable lid. The process begins with a misfolded protein entering the GroEL chamber. The chamber provides a hydrophilic environment, contrasting the crowded cytoplasm, which encourages the protein to fold correctly. GroES then binds to GroEL, capping the chamber and creating an isolated environment. Within this protected chamber, the protein is free to explore different conformations as it folds. GroEL, fueled by ATP hydrolysis, undergoes conformational changes that further assist in the folding process. Think of it as gently nudging the protein towards its correct shape. Once the protein reaches its correct folded state, GroES detaches, opening the chamber and releasing the now functional protein. This cycle can repeat with GroEL and GroES assisting in the folding of multiple protein molecules. Section 7. More than folding, the diverse roles of chaperones. While their role in protein folding is crucial, chaperones are involved in a much wider range of cellular processes. They participate in protein transport, assembly of protein complexes, and even protein degradation. For instance, some chaperones help to transport proteins across cellular membranes. Others assist in assembling multiple protein subunits into larger complexes, ensuring these complexes form correctly and function properly. Chaperones are also involved in recognizing and targeting misfolded or damaged proteins for degradation. This quality control mechanism prevents the accumulation of harmful protein aggregates within the cell. Their diverse roles highlight the importance of chaperones in maintaining cellular health and ensuring the smooth operation of various cellular processes. Section 8. When proteins go wrong, misfolding and disease. When protein folding goes awry, the consequences can be severe. Misfolded proteins can clump together, forming aggregates that disrupt cellular functions and contribute to a range of diseases. Alzheimer's, Parkinson's, and Huntington's diseases are examples of neurodegenerative disorders linked to protein misfolding. In these conditions, misfolded proteins accumulate in the brain, forming toxic aggregates that damage neurons and impair brain function. Prion diseases, such as Creutzfeldt-Jakob disease, are another striking example. Prions are misfolded proteins that can induce normal proteins to misfold, leading to a chain reaction of protein aggregation and severe neurological damage. These examples underscore the importance of chaperones in preventing protein misfolding and maintaining cellular health. Understanding the role of chaperones in these diseases could pave the way for new therapeutic strategies. Section 9. Chaperones, tiny machines with a big impact. Molecular chaperones, like the remarkable GroEL and GroES complex, are essential players in the bustling world of the cell. These unsung heroes work tirelessly, ensuring that proteins fold correctly, preventing harmful aggregation and maintaining the smooth functioning of various cellular processes. Their discovery, stemming from a serendipitous observation in fruit flies, has revolutionized our understanding of protein folding and its implications for human health. As we delve deeper into the intricate world of chaperones, we gain valuable insights into the fundamental processes of life. Their importance extends beyond basic biology, offering potential avenues for developing new treatments for diseases linked to protein misfolding. Chaperones, though tiny, have a big impact, reminding us that even the smallest components of the cell play crucial roles in maintaining life's delicate balance.